Okay, we're going to have a couple of quick podcasts about cell membranes themselves. We will have no podcasts, well, maybe I shouldn't say that, but cell membranes, as far as a component of a cell, are foundational. They're extremely important. They are what creates the cell as a separate thing from its environment. And they do many, many, many important things for a cell. So we're going to have a special little talk about cell membranes in particular. Okay. Now, functions of cell membranes are many, and you should be aware of them. Of course, they hold the cell together. They hold all the little parts together and keep it as a functional unit. They protect the cell from bad stuff that we don't want to get into the cell or hurt the cell. And that leads to this function, which is to control what comes in, what enters, and what leaves the cell. You know that as being semi-permeable. It's not permeable to everything. It's only permeable to certain things. The things we want to enter are things like nutrients, sugars and things like that, oxygen and other useful stuff we want to come in. We want nasty stuff like waste products to go out so that we don't poison ourselves, and also something that we will talk at least just a little bit about. Cell membranes are the parts of the cells that bump into other cells because they're on the outside. So it makes sense then that those are the parts of cells that communicate actually with other cells because they're the parts that are on the outside and they'll be bumping into other cells and they can have ways to communicate with each other, and we'll see what that means shortly. Okay? Membranes also play a really important role with organelles. Membranes don't just make up the outside boundary of the cell, okay? They make up the inner boundaries too, or what I mean by that is the organelles, the little compartments that are inside a cell. When we drew our cell on the board, we had the outer membrane, but we also had all the little cell compartments in there, and they are all, almost all, made out of membrane material, and they separate, these little compartments separate activities in cells that need to be separated. For example, if, I'm going to draw down here, if this is my cell and I am trying to make food molecules in that compartment, I cannot also break down food molecules in that compartment. Those making food and breaking down food are things that cannot occur in the same exact place. They are incompatible. I couldn't make food and have the enzymes for breaking down food all in the same spot because they would just all end up being broken down. So we separate things like making food in a plant cell from digesting food in that same cell. We separate those processes. That's what organelles allow us to do. Those compartments, those organelles, are basically made out of the same cell membrane stuff as this outer boundary. They're just compartments on the inside. Of course, there are some specializations to these compartments, some certain chemicals that are in them or whatever, and we'll talk about that more later. But nonetheless, fundamentally, it's basically the same material. So when we talk about organelles, we're talking about basically little balloons made out of cell membrane that are inside the cell itself. Okay. Now, what are cell membranes made of? We already mentioned this in class. Phospholipids. Phospholipids. Now, what I want you to notice from this word is that it's a lipid. Okay, and so the first thing that should come to your mind when we think about lipids is this picture that we've talked about many times before, okay, where we are talking about a triglyceride which is nothing more than this thing, which we already know is a glycerol molecule, which is basically just a hook to hook these three really important fatty acids together. Okay? And we know that the lipid we've been talking about, a triglyceride, has three fatty acids attached. Well, if we make a phospholipid, we are going to have one of these go missing. That's not going to be there. And in its place is going to be something called a phosphate group, which we are just going to label like that. We're going to make it look like a little circle with a little P in the center. That will be our symbol for that. So a phosphate group is going to go in place of a fatty acid. And what's important about that is a phosphate group has a negative charge to it. A negative charge. Okay, now I don't know if you'll remember or not, but fatty acids, the thing about lipids is that they are non-polar. Remember my bottle of water and oil. We can shake it up, but still the water and the fat or lipids separates from, the oil separates from the water, because the water, the oil's non-polar and the water would rather stick to other things that are like little magnets, which is basically itself in that bottle, rather than trying to stick to the fatty acids, which have no attractiveness. They're not polar. Well, this has a negative charge, and that means it's polar. It's got it's going to attract something. And if you're water, remember water molecules essentially look like this. The big one's the slightly negatively charged oxygen. The small ones are the slightly positive charged hydrogens. And if you're a water molecule and you see this thing that's negatively charged, your little positively charged hydrogen end is going to want to cling to that. Okay? And that causes some interesting things to happen when we put phospholipids in water. Basically, this is how we're going to draw a phospholipid. It's going to have a circle with two sticks. In reality, this is a more accurate model over here, but that's complicated. So we're going to do this with this circle being the phosphate head and these sticks being the fatty acids. This part is polar. This part is non-polar. And that means it's got kind of a split personality. Part of it, the polar part, is I love water, I love water, I love water. And then the non-polar part is like, what, and doesn't really care about water, okay? Now, there are words to describe that. If you really don't like water, you are hydrophobic. That's all one word, but I can't fit it in. Phobic means you have a fear. Well, that's not quite the case with fatty acids. They just don't really care, but we call them hydrophobic. They don't like waters, theoretically. Polar things, however, love water, and they are hydrophilic, okay? Philic means to love, to be attracted to, okay? Now, the phosphate head is this part, okay? It is hydrophilic, okay? Attracted to water. Sorry, I kind of scribbled over that. The fatty acid tails are hydrophobic. We already said that. Repelled by water, okay? Now, these phospholipids, though, to make a cell membrane, we would have thousands of them, and they would arrange themselves, as we already know, as a phospholipid bilayer, okay? The question is, how do they do that? What causes them to be arranged as a bilayer, okay? Essentially, what happens is water molecules, if we dump, well, let me back out of this for just a second. Let me go to an animation that I've got, okay? If we dump a bunch of phospholipids into water, okay, they are going to behave in a particular way. So let me set this drawing up. This part here is water, and I can't do my pen on this right now, but this in the blue is water, and this up here is not. Okay, now we're going to dump phospholipids in it, and look what parts of them are attracted to the water. Those polar heads, the little circles, are all attracted, which means the tails get thrust away from the water. Okay, now if we shake it up, if we shake up the water and shake up the phospholipids in the water, they will form little droplets just because of the reactions of the polar heads to wanting to be near water. And then by default, the non-polar tails get kind of turned away from water. Well, if we have even more lipids in the water, okay, essentially what they do is they form this bilayer. And what you see here is we've got so many lipids in there that now we've got these guys near water on the outside, these guys near water on the outside, and the nonpolar tails by default get shoved in towards each other. And that's what you're seeing. This is creating a whole big cell membrane right here with the nonpolar heads to water, nonpolar heads to water on the inside part of the cell. And then this relatively dry region where the fatty acid tails are pointing to each other in the middle of that bilayer. Okay, so that is why we get this bilayer shape. Okay, okay. If we mix them in water, the water loves the polar heads. There we go. Here, and the water loves the polar heads, and so it's going to be having access to them here, whereas the tails get kind of isolated and pointed towards each other. Okay, so water is attracted to the hydrophilic heads and not the tails, and so the water pushes the tails away, and they end up pointed at each other and away from the water in a solution. And that's how we end up with these cell membrane structures in water, okay, resulting in this bilayer. Now, that's a kind of a crazy model, but it's a little more molecularly accurate, but it's really the same as what you just saw. This is showing the polar heads in this part, and this is also showing polar heads, and these would both be pointed towards water. This just might be water that is outside the cell, and this is water that is inside the cell. Remember, you've got water in your cells as well as outside of them in your body. That water is almost everywhere. And then the non-polar part is in the middle. All right. And so now that this membrane is going to serve as a barrier, and only certain things are going to be allowed to pass through that barrier. What could pass through this barrier? Well, only things that are either really tiny and things that are essentially non-polar. Why are non-polar things the only things that can pass through? Well, we know that this outside part is polar, and polar things attract polar things. So if I am a water molecule, for example, if I'm water, I'm going to go up and I'm going to get stuck to that because that's polar, and I'm going to love it and I'm going to cling there. If I am a sugar molecule, I am also polar, and I'm also kind of big as well. And so I'm going to come up to that and I'm going to see that and I'm going to stick to it because I'm polar. Plus, I'm so darn big, I'm not going to be able to wiggle through that tiny little spot. But if I am an oxygen molecule, I'm non-polar. And I might just be able to be small enough to squiggle through that little crack and get to the inside, okay? So that is one way important way that phospholipids act as a selectively or semi-permeable barrier. Okay? Now, what can pass through? We just saw this lipid pass through. Why? Because lipids are nonpolar, and so they can usually slide right on through if they're not too big or don't have some other complicating properties about them. Sugar can't get through. It's polar, it's big, it's going to cling to the heads, it just can't get through. Okay? Water can't get through for the same, not because it's really big, actually it's quite small, but it's so polar that it likes to cling to the polar heads. Salts are also polar, they can't get through. Waste products also tend to be polar. So this part, this nonpolar part, and this polar attractiveness makes these things, these wastes, sugars, water, salt, tends to make them impermeable. They cling to the polar part and they can't get through. Okay? So, because of the close placement and the polarity of the phosphate heads, they're close to each other, there are some molecules that simply cannot penetrate, but there are some that can. And since some are small enough and nonpolar enough to get through and some are not, that is called being semi-permeable. Okay? A really important quality. Now, we just saw, I'm going to go back one, we saw that sugar can't get in, water can't get in, salt can't get in. Well, wait a minute. Those things need to be able to get in. So, how are we going to allow some of these things to get in and control when they get in and things like that when we really do need some sugar to get in some of the time? How are we going to do that? How are we going to control it? How are we going to get it in? That leads to a talk about some other things that are part of a cell membrane. Please note that phospholipids, are the foundation molecules of cell membranes, and they are really important. They are not the only part of cell membranes. There are other materials as well that are parts of cell membranes, and these really important things are usually made out of protein, and that's what the next podcast is going to be about. Okay, so I'm going to stop this one and then we'll have another one, and that'll pretty much wrap up cell membranes, I think, at this point. All right, hope you have a lovely evening, day, whatever you're having, and I will catch you in the next podcast.